Hello, good morning. We have a new session in this series of talks in Introduction to Bioinformatics. And today we have a very special day. We have two former students of this degree. And we will start first with Aina and later Dima. So I propose that, well, after Aina's talk, there will be time for questions. So please ask general questions related precisely to Aina's talk. And then after the talk by Dima, yeah, you can of course ask questions related to Dima's talk, and then both of them will come to the stage, and you can ask more general questions regarding trajectory, path, recommendations, and so on. Okay. So yeah, so please, yeah, let's welcome Aina Martí. Aina Martí, I mentioned, is a student of the third promotion of this degree. She finished in 2021. He did a summer internship at the CRG, then the final degree project at the CENAC, the National Center for Genomic Analysis. Did a master's science in data science in the WOC, and very recently she has started doing the PhD in the Wellcome Sanger Institute in Cambridge. The talk today is named Deep Mutational Scanning, from bachelor's degree to lab and data analysis. Welcome.
Thank you very much. So good morning, everyone. So as he said, I'm Aina Martí, and today I'm here to explain you a bit of what I'm doing right now, which is deep mutational scanning, and a bit of how I ended up doing this in the lab and analyzing my own data. So first of all, a bit of my career path. I started doing this same bachelor's degree. I started 5 years ago, and when I graduated, I went straight to work in the Learner Lab at the CRG. But how did I manage to enter this lab from the very beginning? So that's because I did a summer internship at the CRG when I was still an undergrad of this degree. And so this opened me the opportunity to work in this lab when I finished, because they liked how I worked. It opened me this door, and I just reached out to them and said, hey, I really loved what I did in this summer internship, can I come back? And they were happy with me coming back, and so I just joined the lab. And so main take-home message here, do internships. They are helpful. You may not like the first thing you try, but try things, discover new things, and you may find something that you love, and it may open you new possibilities. So when I was, while working in the Learner Lab, I did a master's degree in data science at the Open University of Catalonia, and since it was online, it allowed me to keep working in the Learner Lab, which was a huge opportunity. And when I finished the master, I wanted to do a PhD. So to do PhD, you really need some funding. You need someone to pay the PhD for you, because it is extremely expensive. So, or either you apply directly to the institute that you want to go and they pay it for you, or you apply for external funding. And in my case, I applied to a fellowship from La Caixa, and they gave me that fellowship, and then I applied to the institute, and they accepted me. So now I am doing a PhD at the Sanger Institute in the University of Cambridge, funded by La Caixa. So a bit of the Learner group. That is where I am right now, because in the Sanger Institute, I'm also working in this lab, in the Learner group. So this is the people in the lab, all amazing people, super nice, all super helpful. And in the corner, there is Ben Learner. He's such a nice boss. So this lab is running between the CRG here in Barcelona and the Sanger Institute in Cambridge. And I was before in the CRG, now I am in the Sanger Institute. So basically, the main thing that I want to highlight right now is that this lab allowed me to work half in wet lab, which is what we call when you're in the lab with pipettes and all of this, and half computational, that is what we call dry lab. This is what I wanted to do. This may not be what you want to do. You may want one thing or the other, but this is my example, and a bit for you to see what it looks like. This is a bit my life right now. So it's 50% wet lab, a lot of experiments, huge experiments, late nights in the lab, things not working many times, but things working super well many of the other times. So it's super nice. I really love it. Then I also have this other half of the computational side. In this case, it is about writing scripts, analyzing data, making plots, checking things that the data that you generate in the wet lab. So this is what my life looks like right now. So in the learner group, we are people from all over the world. So the United States, Colombia, South Africa, Germany, Italy, and many places. And we also studied very different things. So for example, chemistry, I did the bioinformatics degree. So we really studied different things, and we all ended up in this lab, working all together. And what we study are proteins. So I guess you may already know, but proteins basically come from the DNA that you have inside your cells. This is the information that you need to function as a human being. So the DNA goes to RNA, then chain of amino acids, and these are the proteins, the chain of amino acids. And they fold in very specific ways. So you can see proteins as like puzzles that interact between them. They need to have very specific shapes so that they fit between them very perfectly. And this fold is what gives them their function. So what do we study about proteins? We study mutations. So mutations are basically changes in the DNA, which logically make a change in the RNA and then make a change in the chain of amino acids. And also there's a change in this structure. So how is every of these mutations affecting this? Is it affecting the way they function? Is it affecting the way that this fold is done? Is it changing its shape? Is it changing its ability to bind to another thing? So this is what we do. We do many mutations and we quantify how are these consequences. So how we do that? We do that using a method called deep mutational scanning. It is a very general method, but I will explain now exactly how we do it in the lab. So we basically synthesize all these sequences. These are DNA sequences. And we make these changes, all possible changes in the DNA sequence. And once we have all these DNA sequences, we put them inside. Inside the cells. And each of these cells will have one of the DNA sequences. So when each of these cells has the DNA, then we make them grow under a selection media. Okay, so it's like a liquid that is quite toxic for the cells. And in this case, we call it selection because we make a selection of these sequences in this way. So if the cell, so if the cell has a variant with a change that is able to fold, the cell will be very comfortable in this medium and it will grow. It will be very comfortable, it will make copies. So otherwise, if the cell has a variant that is not able to grow, that is not able to fold, then the cell won't be able to grow. So this is the type of selection that we do here. So what will happen if we let them grow for some time? That the frequencies of these cells will change. Okay, so see the green cells. They went from 2 to 5. What does that mean? That it was able to grow in this selection media. So that means that that this specific variant with the TA change was able to fold. So with this, we know that this specific variant was very strong in this selection medium, so we know that it folded. But what happens in the case of the purple ones? That they completely disappeared, they died. So that means that the DNA that the purple cells contained was rising to a protein that was not able to fold. So then this is also very quantitative because we don't have just fold and not fold. We have a very wide range of effects. We can have like an effect of 1, an effect of 1.02, and etc. Because, for example, these cells, these purple cells went from 2 to 0, but we also have the red ones that went from 2 to 1. So this is kind of a middle step between the green ones. So we have many different type of ranges in here. So the mutational scanning is basically this method that tries to compare these frequencies between the beginning and the output. So input and output. But what happens in the middle can be very different. You can choose to use different medias, or maybe not do it inside cells. Maybe do it in vitro. So there are very different types, and you can select for folding, for binding, for function inside the cell, or whatever. So, okay, so what we generate from these experiments is this type of maps. In the x-axis, you have all the amino acids in my protein. In the y-axis, you have all the possible changes that can be done to each of these possibilities. So imagine the first one, serine. What would happen if I change this serine for a proline up there, the P? That I am killing the protein. I'm making it not able to fold anymore. But what happens if I change the serine for an alanine, the A at the bottom? That I am kind of actually improving the protein. So this is what we do for all possible mutations in a given protein. And the good thing about this is that in a flask with that media, we have millions and millions of cells. So we can quantify many, many of these mutations, not only this, but we can generate many maps in very few experiments. So this is a super powerful method. So in my own project, what I am studying are PDZs. So you can apply the previous method that I said in many different proteins. And I am applying it to these ones. I'm generating the maps for this specific protein. These are protein-protein interaction domains. First, domains means that they are a smaller part of a bigger protein. They are a small part. So for example, in this case, the hand of the blue protein, for example. And in protein-protein interaction means that they basically make interactions with other proteins. So you may think, okay, this is a very simple function, not very important, but it is actually super important. Why? So this is our usual perception of a cell. This is what we have all been told from the very beginning. This is how everyone draws a cell. But this is not actually how it looks like inside. This is a more real representation. It is completely full of things, completely full, like many, many proteins, DNA, many things inside. And so if these two proteins need to be together to function, once they find each other, they need to stick together. If they don't stick together, they will get lost in this ocean of things inside the cell. So that's why PDZs are so important. And actually, this is quite a general function, no? It's just joining two things together. So they are important, but also present in many different biological processes. What does that mean? That if they fail, they get implicated also in different diseases all around the body. So for example, cancer, cystic fibrosis, Alzheimer's disease, Parkinson's disease, and vision and hearing diseases. So in medicine, we want strategies to turn on and off proteins as we wish in order to cure these diseases. Because when a protein is not working, it may be because it's working like crazy, it's functioning a lot, and you want to turn it off, or maybe because the protein is not doing anything, and you want to turn it on. So that's what we want in medicine. We want ways to turn on and off proteins as we wish. So one of these possibilities is allostery. There are different possibilities, but one of them that is recently being very studied and very promising and etc. is allostery. What is allostery? So you can imagine the interaction of two things as a puzzle, no? Then they have a very specific shape that needs to fit together very well. And if you do a mutation right in the region where this binding is happening, you can expect that this is not going to bind because the shape has changed, no? But what happens if you make a mutation very far from what, where the binding is happening? I mean, you would not expect this to affect the binding, but sometimes it affects. Sometimes it increases the binding, sometimes it decreases the affinity of binding. So we want to understand that. This is kind of a domino effect. You make a change in one side, and there are changes propagated throughout the structure so that it actually makes an effect in the other side. This is a loss theory. When building our maps, we want to find these positions that when we mutate it in the other side of the protein is actually affecting this other side. So it's affecting the binding of the protein. So that's what I am studying in these PDZ proteins. They are known to have these allosteric effects, and we want to find these positions far, so where we can kind of build, well, not we, but we generate this knowledge. We can build a key that is used in the other side of the protein so that we can try to turn it on and off, but not directly in the binding interface, but outside. So this is providing an alternative to what usual medicine drugs do. Okay, so this is my project, but now a bit of an overview of why we do demutational scanning. So as I said, you can apply this to many different proteins. I'm applying it to PDZs, which are protein-protein interaction domains. You can also apply it to oncoproteins and different types of proteins that might be of interest to you to cure some disease. So when we do this work, we are building the knowledge. We are building the foundations. We are creating these maps of effects for many different types of proteins. But not only that, we also try to understand what's happening. We build machine learning models, biophysical models. We do things to really understand what is happening there, understand the protein, and maybe try to generalize that knowledge to other proteins. But we also keep building and building data, so that at some point, we will be able to train predictive models. What does that mean? You make an algorithm that takes many examples, and it learns from them. And learning from the examples, then you can actually apply the knowledge learned in the examples to another thing. So obviously, the more examples you give it, the better it will understand what's happening, and the better it will predict to another thing. So all of this knowledge and predictive algorithms and all of this drives us to be able to speed up personalized medicine. Personalized medicine is basically when a patient comes and has a cancer that has a specific mutation. Well, maybe not only one patient, maybe different patients. But you want to target that specific protein with that specific mutation, and you want to turn it on or off depending on what the problem is. So this is personalized medicine, and understanding how all these mutations work, understanding what is happening there, it brings us to personalized medicine. So this is the overview. This is my example of what I am doing. I'm doing a PhD, but you don't really need to do a PhD if you don't want. You don't really need to do a master if you don't want. But this is kind of the typical path, I would say. But there are many paths to go to where you might want to go. So I guess this is everything from my side. Hope you understood it. If you don't, please ask me.
Okay, thank you very much, Aina. Very clear presentation, very nice. Time for questions.
Okay, how did you apply for the internship, and what was your motivation for doing it?
Actually, I applied just because I knew there was an internship. And honestly, I was so shy that I actually didn't want to go. I just went because I felt like I had to at least apply. Then they accepted me in the internship. It's basically like a form that you have to fill, like your grades, your name, why you want to be there. In my case, it was because I want to try things. I want to, like, at that moment, I just did 2 years of the bachelor's degree, so I wanted to go to the lab, try things, and this was my motivation. They accepted me, and I just went. And it turned out to be the best idea ever, and now I'm here because of that. So that's why you have to try things, even though if at this moment you may think I'm not good enough, or I won't be able to do that, or I just don't want because I'm shy. I mean, I understand all of these thoughts, but you still have to try.
So you told us you did a master's in data science. How can that go with doing half work, like work 50-50 in a wet lab and a computational?
Yeah, okay, so I did a master's in data science that was very general. It was not specifically focused on biology, but when I finished the degree, I went to this lab. I started learning on all of the wet lab techniques, because in this degree there's not many wet lab things, but there I learned all of that, and when I sit it in the computer, I was like, I don't know what to do with this. So I felt like I needed to deepen my understanding on how to analyze all that data, because at the end, it doesn't matter what you do in the wet lab if you're not able to analyze it. No? So in my case, since I wanted to really understand all the statistics behind all of this analysis, neural networks, all of this, so I wanted to deepen my understanding of that. And obviously, that's bringing me to better analyze my data that I generate in the lab.
Any limitations in deep mutational scanning, and what is the key technology used?
Limitations, you mean?
Yeah, in deep mutational scanning, and what is the key technology used on it?
What do you mean about key technology?
Like, as you said, the algorithm to know the personalized medicine. So what's the technology behind it?
Okay, so limitations. Yes, it has some, because obviously we would like to do as many variants in one single experiment as possible. This is already huge and much better than previously doing one by one. But limitations. I guess I would like it to be more high throughput, meaning more variants at the same time. And also, we have an assay that, as I said, works inside the cells. It doesn't work for all of the cells, all of the proteins, sorry. So maybe if it's a huge protein, it's being biased towards being more difficult to fold. So there are some things that maybe we don't fully understand yet, because we're working on it. It works super well, super specific on many of these proteins, but it is not applicable to some others, let's say. So this is one limitation. Also, we would like it to be more high throughput, etc. And the technologies. Okay, so once we get these grids, so the frequencies in the beginning and after, we apply statistical methods. This is not like a technology itself, it's just computations, formulas, and all of this to compute the exact frequency from the beginning versus at the end. Then with this, you apply neural networks, which are basically algorithms that you give it examples and it learns from them, and it trains inside of it some things that you can then just understand what's happening. But this is to understand biophysically these differences in frequencies. Because it doesn't matter that you go from 2 to 5 if you're not able to give it a biophysical meaning with some units used in biophysics and etc. So, yeah.
What would you recommend? Sorry. What would you recommend to yourself if you were at our position?
Trust more myself, I would say, at that point. I had a huge imposter syndrome, which I still have. I would say that. I would say two things. Like that internship, I know I did it, but I was this close to not do it. So do it. Don't think about it. Do it. And if it fails, it fails. No problem. Like if you end up not liking it, then try another thing. That's not a problem. I liked the first thing I tried, but it might not be the case for everyone. But just try and find your place and just trust yourself. You can do things. Even though you think you are not able now, I did that internship after the second year, which you would think you still don't know enough, but I knew something, and they saw it. And they, after the third year, they allowed me to come back and work with them. So trust yourself.
Okay, thank you very much. Thank you very much, Aina. So now time for next former student, time for Dima. Okay, Dima is from the second promotion. Dima finished in 2020. Also did the final degree project in the SENAC CRG. Did the same master in the WOC, data science, and well, at the same time, he also was doing work while studying. So he started working at Omniscope as a computational biologist, and he continues to work there. Yep. Thank you very much. The talk is going to be immunosequencing from a blood vial into a diagnostic and therapeutics. Welcome.
Yep. Thank you, Arnaud. So first things first, I cannot explain you that much of what I'm doing for some NDAs and protection stuff since I'm in a company, it's not a public institution, so some things I just cannot disclose. But anyhow, I'll try to give you my perspective on how I went from your position into where I am now. It was like a 5-year journey, a fun one, and this is where I ended up at Omniscope. So, does it work? Okay, my bad. Yep. So for some reason, the PowerPoint messed up for going from Ubuntu to Windows, but anyhow. So first of all, as I now mentioned, I'm the second promotion, so I'm the second year that got into the university. When I was doing this degree, it was very different to what you're doing from what my friends and other teachers told me. So I started as a bioinformatician, then once I finished the degree, before I finished, I did my final grade project with Oscar Lau, and I went from not knowing what I wanted to do to seeing that I love evolution. I went into evolution, I did my final grade project, I got very good score, I enjoyed it a lot. But the whole point and the issue was that evolution is fun, it's a very interesting subject, but it just doesn't get funded as much because it's very hard to apply it to anything that gets you benefits, so anything that is related to medicine or something personalized, because it's just research. It's not a research that goes into industry or goes into a product that you can later get profit from. So that was sad because I really liked it, but I saw that, I couldn't stay there to do publish a paper or anything, so I had to do something different. So I did the same master's as Aina, so I did data science. It's very general, and it was nice because it teaches you, the whole difference between the degree and the master's, in my case, was that degree was fully focused on biological data, and everything was did with biological data, and I wanted to see how well can I apply what I learned from biological data to anything that is not biology related. And it turned out well, so working with images, text, speech data, and many other things, but once you start in biology, you end up in biology. So my master's thesis was about lung cancer and identifying the area where the lung cancer is related in the CT scans. And I ended up in another biological company, Omniscope, so it's a startup, it's a very young company. And I had an interesting journey with the company because when I got the interview with Holger Hein, which is the scientific head of the company didn't exist. So when I didn't apply for Omniscope position, I applied for a position at the CRG because Holger is at the SENAC CRG. And I did an interview, and all of a sudden he told me, okay, there is a company where you're starting right now, do you want to join? I said, okay, I'm joining. And here I am now. So first 2 people from my team that joined was me and Juan Luis. So we built the ComBio part of the company, the 2 of us, and now it's all of these people, lovely people that I enjoy working with, and we're still a small team, but we've done many things, and I'm going to explain a bit more what do we work in and with. So the idea of modern medicine, it seems to be, it's really hard to define. What is modern? What is a universal thing? How can you create a tool or how you can create a diagnostic that can be universal for everybody? it's very hard because each person is different, but we do share some similarities like the body and different immune systems and so on. So the first universal diagnostic tool we can call the X-ray in 1895. What is the problem? The problem is that it's costly. Like it used to be costly, now it's much cheaper. You can do it anytime, at any day. And if you don't have something universal like an X-ray that you can scan anybody, any part of the body, the way of diagnosing something or identifying an issue that you have can be really costly and can take too much time. So now going from X-rays, we want to do something that is more related to the diseases themselves. So we have an immune system, all of us, and this immune system is the system of your body that works against all the different antigens and diseases that can get into you. To sum it up, I work with the adaptive immune system, like generally Omniscope, and you have two types of immune cells. You have the T cells and the B cells. And it's very nice that I'm not talking about proteins right now because I'm going to talk about proteins myself and the bindings and everything. Basically, you can have, like the T cells and B cells, they have the cell itself, but then they have the receptor. Like this part right here is the receptor. And the whole idea of having the specific shape or like specific composture that you can bind to something else also applies in these proteins, in the receptor. The receptor is like a set of different proteins that have their sequence, and from the sequence you get the shape. And the important part of the immune cells is that each receptor can work against multiple infections or diseases, viruses. There are many things they can recognize. For example, the T cells, you have 95% of the T cells have alpha chain and beta chains. The smaller 5% have gamma chains and delta chains. But the important part here is that you have the variable region, and it's like the key component of the T cells. Why? Because as you know, you can get a lot of infections, a lot of diseases, and as you can imagine also, all of these diseases are unique. Because if they weren't, if all of the diseases were the same, we wouldn't need to have a variable region. We would have one constant region that would go against everything. But that's not the case. So the alpha and the beta chain, they both can vary, and by varying the sequence, they vary the shape, and thus you get a system where one T cell can recognize a multitude of different diseases and can work against them. And there are different subtypes of T cells. You have the killer T cells, the helper T cells, the memory T cells. I don't want to bore you way too much about that, but the general idea is that they either kill not the disease, but like the cell that they find, or they help the B cells to work against them. Because the idea for the B cells is very similar, but instead of having alpha and beta chain, they have the light chain and the heavy chain. And the B cells are the ones that activate the antibodies later on the cascade. Nice. And what I lacked when I started the degree and when I was doing this subject like 5 years ago was to understand what is the connection, or how do you go from wet lab, or from what we know as science nowadays. Because when I went to school, when I thought about science, I thought about pipettes, I thought about like liquids and just being in the lab with the white trench coat. So what I want to make you understand is that as a bioinformatician, like for example, Aina, you can be at any of the 3 steps here. Me personally, I don't work at wet lab, mainly because I don't want to spend way too many hours at night in a lab doing experiments that they can fail or not, you never know. I'd rather be at home with my laptop, with my cat, just working on a sofa, and that's very nice for me. Other people like other things, and that's totally valid. So first things first is I'm going to give you the example with the T cells, because we have 2 assays, the OST and OSB. So OST, you get a blood draw, and with that blood vial, what we do, we sequence we say we sequence the T cells, but it's not really true because you cannot sequence a cell. What we do, we sequence the receptors, and And those receptors are composed of the VDJ. So VDJ are three different genes that when joined together, you get the entire part, like entire receptor of the T cell. And to put in perspective, the earlier technologies would be able to sequence 1,000 cells, 10,000 cells, or 100,000 cells. What separates the OSC from the others is that we can sequence up to 1 million T cells. And it's a lot of data because it's only one sample. So as you can imagine, getting that many cells per sample, imagine you have a cohort of 100 patients of like pre-treatment, post-treatment. That's 200 samples that you multiply by 1 million and you get a lot of data. What we need is like a place where we can put the data and we can analyze it, which in this case is the OSIP, or Omniscope Human Portal. And this is the place where the data is stored. You can open, you can access it. I'm going to show it a bit later in the presentation. And where we apply AI algorithms to retrieve information from the data. As example, Aina said, you can apply neural networks, you can apply different statistical algorithms for that. And once you have the data on the IP, there's another thing that is Omniscope intercept. Like generative AI or Gen AI, I guess everybody has used ChatGPT at some point of this degree already. I did in my master's degree, so don't feel bad about it. So what we do, we use Gen AI to work with all the data that we generate to generate even more data, so we don't have to sequence, we don't have to like extract blood, but we still can get more data to work on and to validate. And here's a more visual presentation which I just explained. And what I want you to understand is that as a bioinformatician, in my case, I work from here onwards. But you should do what you like. And I also feel the notion that in the degree we're kind of forced to do something all the time, and we are forced to say, okay, I like that subject, I want to do internship in that. And I don't want you to feel this way. What I suggest in your position, especially in the first year, is to try and see what subjects you like. And based on that, try yourself in the wet lab part, or try yourself in the wet lab plus the data analysis part. Or if you can, or if you want, just learn more about the AI, the machine learning, and try yourself only in the AI area of the bioinformatics. It's a such like big field that you can explore. You can probably find something that suits you perfectly and you actually enjoy doing it and it doesn't feel like work. Like that's what you should look out for in the degree when you finish it. So with the data we generate, we can go into high-value diagnostics and informational therapeutics or personalized medicine. So all of this can be summarized as personalized medicine. So that's what I mentioned earlier. Other technologies go not as deep. So they didn't really need anything that is on such a level of complexity that you need to use AI models or AI algorithms to work with the data. You can just do very simple statistics stuff that works equally fine. But in my case, on daily basis, I do develop models. Again, I'm not allowed to talk about them too much. That's the sad part about working in the company. But on like day to day, I work with AI. I work with ML. I'm trying to make sense of those 1 million T cells that we find, because you can imagine it is like a CSV file that you open. You have 1 million rows. And if you don't apply anything that can automatize the analysis, you won't spend your day going row by row, seeing what is the sequence, what it works, matching that with databases. This has to be done in an automatized fashion. And here's an example of like why do we need 1 million T cells per sample? Like T cells, B cells, doesn't matter. Both are needed. Is basically it's not a one-to-one relationship, as I mentioned before. So you can have multiple T cells working with multiple antigens. And to give you like a good example of why this is so complex, imagine labeling a picture that has a cat on it. It's very simple. It's one picture, one cat, you just label it cat, done. imagine how hard it is to label a picture of a zoo, where you have a multiple number of animals, and they're mixed and mashed all over the place. So it's very hard to put a label on something that is so mixed. And as you can see here, when you're getting a sample from a patient, and you want to see if this patient has some sort of disease, let's say influenza or COVID or something else, it's not as trivial, because you can have a subset of cells that might work with COVID, other subset of cells that might work with influenza, and some cells might be memory T cells that come from infection that you had a year before the sampling, or they might be the ones that are reacting now. Like, there's so many things you have to take into account, that the more cells you have, the more a good overview of the actual immune system of the patient you have, and the more precise would be the diagnostics. And as we know, the better the diagnostics, the easier it is to treat the patient correctly, and the higher it is the probability that what you're doing will actually help somebody feel less pain or just be healthier. And this is the example of the portal. So from everything I'd explained up until now, I work with each and every part of the company. So also to show you that being bioinformatician doesn't mean you work with one thing and only. You can have multiple projects, you can have multiple things you can work on. If you're working in a public domain, like Aina, usually you have one project that you're attached to, and you and another person, like the head of the lab or somebody doing PhD, are the 2 people or 3 responsible for the project, and you kind of like focus all your time on that. Maybe some other smaller things that you can work with, but if you go into the industry, unless you go into like a big company that require one person for that specific thing, you usually end up working with many things. So here you can see the whole design of the portal, and I also took part into like designing it, and in the analysis part, the network analysis, database match are the and even the sample summary and comparison. Like all of the 3 notebooks are the ones that I dedicated a lot of time on, and I enjoyed doing. Why is it repeated so much? Oh, and here's the more AI-related stuff. I'll try to talk as much as I can without getting fired, so bear with me. A quick summary of what AI is, or like machine learning in general. You have data, you have a model that gets the data and produces either more data or labels what you're giving to the model, or like cluster data. There are many different algorithms. In this case, this is an example of generative AI. So Gen AI with text or images is very simple. You input prompt, you get an image, or you input text, you get more text. With this, it's a bit more complex, because how do you generate T cells? Like, it's very not trivial question to answer. So the simple thing is you get the 1 million T cells, 1 million receptor that you have. You create a Gen AI model that I cannot elaborate on. But basically think of it as working with text, like similar to ChatGPT, but not exactly. You give it a sequence, like a set of sequences, and with the set of sequences, the Gen AI in this case try to create new sequences that are similar to the ones that you've given, that for example can be validated by the wet lab, that they are related to COVID. And the Gen AI, what it does, it gives you even more sequences that might be related to COVID for you to use them later on for diagnostics, or if you do it for cancer, for example, you can try to generate sequences that might work for that specific patient based on the immune system that they have, to try to do the immune therapy, to give different T cells that try to work against the cancer. So it's a very cheap way of, even if you don't get samples or you don't get blood, you can still generate data that you can validate and work on and try to help people with it. And so yeah, that's like a good summary of what I explained, or what I can explain. You get high quantities of data, and it's just unreal to work with the data by yourself. So you have to develop models and develop ways to go from that simple sequences to something you can interpret in a medical context, which is also not easy. And once you have that, you can try to use generative AI to, instead of going for more patients and trying to find more cohorts, because sometimes it's not that easy. Some people, like, it's not that easy to find cancer samples from like later stages or from different parts of the body. So it's much easier to get Gen AI for you to do that and get the data from it and try to validate it this way. So yeah, if you have any other questions, you can email Omniscope. I'll answer them happily later. But now if you have any questions for me, feel free to ask, and I'm more than happy to answer.
Okay, thank you very much.
You said during the bioinformatics course that you were more inclined towards the biological features of this field. It seems now that you are more interested in the data analysis side. I was wondering if you could give me a pro and con from both experiences, maybe?
Yeah, so the good part about being focused on the computational stuff is basically that you do not depend on the location or time. You can work from home, you can work from an office, you can work during the day, you can work during the night. You're not limited to something like a lab. And that's like the biggest con of being in a lab, because you have to be there at specific hours, you have to wait out 2 hours for the experiment to finish, and you cannot just go home at any time. So it binds you to a location, and that's something that I personally don't like. But also because I did do like a small internship in the first year in a wet lab. The satisfaction that you get when like experiment runs well and fine is very, like, it fills you a lot. It's a fulfilling thing. So it just depends on you. If you like working with your hands physically, wet lab all the way. If you don't want to be binded to somewhere, just do anything that's related to data stuff, data analysis, machine learning, even biostatistics, all of that can work fine for you.
Do you think that artificial intelligence is the technology that will make like the actual science make a big jump, and why?
It's already making jumps, and it's been making for a while. From my experience, what we've done at Omniscope and my personal projects, big overview again, I don't want to get fired. Taking the blood sample, transferring the T-cell receptor information into a different data structure, and using that data structure to classify the patient, I think it's one of the biggest jumps you can make, because what this can do is, if you get, simple example, COVID, and you get a blood sample early, if you can detect that your immune system is working against that specific antigen extremely early, like first week or second week, you can already put the patient on treatment much earlier than you can physically see it. And that's even better for cancer. If you can detect that somebody is developing a cancer much earlier than second or third stage, you can put them into therapies that are not as harsh as like radiotherapy and chemotherapy, and still combat it and make that person's life easier and just not as bad. And there are many other fields where this has been applied. Again, the idea why I did my master's thesis on lung cancer and scans, because I've seen other algorithms that can detect the shape in a 3D area. So you scan you look at the images, because when you get a CT scan of a lung, it's like slices. So if you look bottom down and you get slices of your lung, and some models are capable of looking at all those images and creating this 3D shape of the lung cancer in your body without the need of a doctor or surgeon opening you up and looking at it. So there are many jumps that are already happening, and I'm just expecting more, if anything.
So at the beginning, where you were presenting, you said that you changed from Ubuntu to Windows. Is there any reason?
Oh, no, it's the presentation. Oh. Because I mean Ubuntu, and you saw some things overlap. Wow. That happen in uni to me all the time. Happen again. But yeah, again, talking about that, learn as much Linux as you can. Try to be proficient in Bash, proficient with anything that is Linux or like Unix in general related, because all the big, even cloud services or like clusters, anything you're going to work with, are Unix based. Do Bash, learn Bash, please.
Or maybe, yeah, Aina, if you could also, you have questions, general questions regarding path recommendations? No? No question? Okay, any final advice that you would like to give to them?
I don't know, like, maybe I would say there will be many subjects that you won't like in the degree. Just work on them. They will be useful at some point. Like, maybe if you end up doing full wet lab, maybe the computational side won't be that useful, but for my example, at least, that I'm doing both sides, everything has been useful, like, even if I didn't like it. And maybe, for example, I used to say that I hated proteins in the degree, and now I'm fully working on proteins. So there are many things that can influence you on liking something or not. Maybe your team, your teacher, whatever. But just give them a try. And I would say that's my advice.
Don't be afraid to try things and to explore yourself. Like, bioinformatics is such a wide area of research. You can go into medicine, you can go into genetics, you can go into proteins, like both of us, or you can fully go into AI. You have so many things to explore that it can be overwhelming. And for this reason, try to find a teacher that you like, that you connected with, stick with them, and they'll help you to guide you on. Because I did my final grade project with Oscar, because I liked the way he taught us the subject, like the subject that he gave us. And then when I joined Omniscope, Holger knew that I did the final grade project with him, so he sent him an email, and basically Oscar told Holger that, yeah, get the guy, and now I'm happy in the company, and I've been there for 3 years. So getting networking and being nice to people also help you on a long run.
Yes, like you took 2 different paths, and I wanted to ask, like, is there a big difference of being in a private company and like a public organization? Or they're more or less the same?
I think they are very different, but I guess you can answer this better than
I've
me.
been in both, and they are different. The feeling, like the biggest difference that what I mentioned earlier, when you're doing something in a public institute, you get one project because you have money that is allocated to that project that you have to spend wisely and overrun on time. In a company, especially like in my case, I'm in a startup, so we're not that many people, so I get more responsibilities than in a usual company. But you get more, it's not like freedom, but you're involved in many things. Like everything that I explained, I was involved in all of that at some point. And the same thing is true for deadlines. You usually have deadlines, but they can be more flexible because you don't depend on other institutions accepting your paper or not. And you can actually work on both. So I think next week or this week, there'll be like a publication in Nature Comms, like Nature Communications, that me working as a company, I still get into the publication because we did a collaboration with a public institute that work in. So if you like both things, you can work in both things. Try what you like. Again, it's a bit harder to get into a company for internship because companies usually want people for with already defined specialized areas and stuff. But try it. If you get lucky, that's great.
Okay, so thank you very much, both.